Hello, my name is Lindsey Jung, and today I'll be presenting my research project titled Strategy for Identifying Potential Cannabinoids Type 1 Receptor Ligands Virtual Screenings Using a Structure-Based Pharmacophore Model. My work was done at an industry laboratory. Today, marijuana is used for therapeutic and recreational purposes. The CB1 receptor is the most commonly expressed G-protein coupled receptor in the brain and is the target for many endocannabinoids in cannabis. These CB1 receptors are found in the central nervous system and are an attractive target today for novel drugs because the binding of endocannabinoids to CB1 receptors play a role in pain relief, memory, mood, appetite, sleep, and inflammation. Cannabis-derived products help treat nausea and vomiting from cancer chemotherapy and relieves pain from damaged nerves. There are also recent reports that cannabinoids such as THC and CBD slow growth or can cause death in certain types of cancer cells growing in lab dishes. However, the legalization of cannabis is controversial due to its psychoactive side effects and addictive nature, which can lead the user to become dependent on the drug. Although some states legalize the use of medicinal marijuana, cannabis is still classified as a Schedule I controlled substance under the DEA. The American Cancer Society supports the need for more research on cannabinoids for cancer patients, recognizing the need for more effective therapies. Therefore, I decided to research potential compounds that can be used as synthetic agonists that bind to the CB1 receptor. The goal is for these compounds to perform the same function as THC and CBD, but without the addictive nature. A pharmacophore is an abstract description of common molecular features characterized by a certain biological macromolecule. Therefore, developing a pharmacophore is crucial in drug discovery projects because you can utilize a pharmacophore to search for a desired drug with certain properties, whether that's through virtual screening, de novo design, lead optimization, multi-target drug design, activity profiling, target identification, and many other types of applications. Many pharmacophore profiles have been published for compounds regarding CB1 activation already, but these are ligand-based. A ligand-based pharmacophore is developed based on hypothetical binding sites because the structure of the ligand-protein complex is unknown. However, a receptor-based or structure-based, also known as crystal-based pharmacophore, gives us all the structural information, proved by experimental data and X-ray crystal structures. Unlike a ligand-based pharmacophore, a structure-based pharmacophore shows how a ligand is binding to a receptor and what chemical feature of a ligand is binding. This renders a ligand-based pharmacophore to be less credible than a structure or crystal-based pharmacophore. The purpose of my study is to search for top compounds and use scaffolds for agonists of CB1 receptor by developing a crystal-based pharmacophore, which has definite binding sites, proving this pharmacophore to be more credible. Figure 2 shows the structures of CBD and THC, which are the most common endocannabinoids in cannabis. Figure 3 shows structures of CB1 agonists that were published. I used these in this project as a template to develop my pharmacophore. Although only 2D structures are shown here, I can hypothesize that all the compounds can have both hydrophilic and hydrophobic interactions, because the compounds have oxygen and or nitrogen. The bottom oxygen atoms in the first two compounds in Figure 3 can theoretically be hydrogen bonding acceptors, and the hydroxyl groups can be hydrogen bond donors. The amide groups in the third compound can generate hydrogen bondings as well. The compounds in Figure 2 are cannabinoids, and the compounds in Figure 3 are 3 known agonists of the CB1 receptor. This shows that there are many different possible conformations for a CB1 agonist. This leads to the question, what are the common properties to ligands allowing it to act as a CB1 agonist? So first, I went to an online protein databank to search for available crystal structures of a CB1 protein ligand complex. I found 3 crystal complexes and visually discerned the binding sites of each. Here's an example of one that I used. It is important to note each specific amino acid that is bound to the ligand because I will need to superimpose the 3 agonists later. As you can see, this dark gray ligand is bound to the receptor. Theoretically, as shown in the 2D structures in the slide before, these compounds can be hydrophobic or hydrophilic. However, the actual crystal structure complex shows the real binding mode, as shown here, that there are only hydrophobic interactions. This is because the CB1 receptor itself is hydrophobic. An experimental approach is more credible than a theoretical one, and my pharmacophore is based on actual experimental data of crystal structures. In order to superimpose the 3 crystal structures of CB1 agonists, I first isolated each ligand in the receptor and then matched the 3 proteins together. As you can see in Figure 6, here are the 3 ligands and these are the amino acids. The entire methodology is done using the SYBL 2.1 program. By superimposing the three agonists, the common features of the agonists can be analyzed. The bonding in the superimposed structures show that three hydrophobic centers at specific distances are required in order to have a binding affinity for a CB1 receptor agonist. I developed my pharmacophore upon this finding using the Unity program. Now that I know the specific factors characterizing agonists as potential CB1 agonists, I can use this as a determining factor to screen compounds. This is my pharmacophore with tricyclic hydrophobic centers. The program that I used revealed the hydrophobic centers to have to be at a distance of 3.99 plus or minus 1, 9.16 plus or minus 1, and 6.95 plus or minus 1. Here you can see the three superimposed agonists in the cavity of the CB1 receptor protein. And here's the pharmacophore inside the cavity. As you can see, both fit inside the cavity. Because the pharmacophore fits inside the cavity, that proves that all the compounds filtered through the pharmacophore search which will be conducted later, will also fit inside the cavity. In order to boil down to the top 20 CB1 agonist candidates, I first screened the compounds that fit the pharmacophore using Unity and the Maybridge database of 54,298 commercially available compounds. Next, I calculated three scoring functions, Chemscore, Gold Score, and Doc Score, in order to confirm the strength of the binding affinities between the compounds and the CB1 receptor. So, scoring functions are mathematical functions that are used to approximately predict the binding affinity as well as the ligand pose between two molecules after they have been docked. Although the compounds that I now have will fit the pharmacophore and fit the binding mode, it is still possible for the molecular structure of the compounds to be larger than the cavity and protrude the cavity, rendering it a defective CB1 agonist candidate. So after filtering the compounds with the scoring functions, I will visually analyze each of the remaining compounds to make sure that the compounds do not protrude any part of the cavity. Figure 10 shows the top four compounds fitting inside the cavity with the common features of the pharmacophore, shown here, these three blue circles, which is my pharmacophore, and these are the three tricyclic hydrophobic centers at specific distances. Here are the results of the compounds after all the docking scores were calculated. The first three rows give the scoring functions of the three known agonists that I first used to develop my pharmacophore. Because I know that these three are already known CB1 agonists, I can rely on their docking scores to find which compounds had the most similar scoring functions. I filtered all the compounds to find the top 20 with scoring functions closest to the scoring functions of the three agonists. If the numbers are close, that means that the binding modes of the compounds are most reasonable to be CB1 agonists. I analyzed the molecular structures of the top 20 compounds, and they are all nonpolar and hydrophobic. The ligand is in orange and my pharmacophore is in yellow. You can see that in these four compounds that I've shown in figure 11, all the compounds fit my pharmacophore. Using common properties derived from three superimposed crystal CB1 agonist ligand complexes, I developed a pharmacophore with tricyclic hydrophobic centers at specific distances. I narrowed down 54,298 commercially available compounds to the top 20 after performing virtual screening and calculating scoring functions. Because I took commercially available compounds, this means that my top 20 compounds are all available in the market and previously used for drugs. Virtual screening and scoring functions ensure that the compounds fit the pharmacophore and are predicted to have reasonable binding positions and affinities with the CB1 receptor. In conclusion, developing a pharmacophore proved to be an effective strategy for identifying potential cannabinoid agonists of the CB1 receptor. 20 compounds were successfully identified through virtual screening. Regardless of whether these top 20 compounds act successfully as CB1 agonists, this pharmacophore developed will still remain useful for other scientists to use for further projects relating to the CB1 agonist and/or receptor, since my pharmacophore is based on actual crystal structures. The properties of my pharmacophore are definite and reliable. Additionally, I contacted a professor that agreed to test the 20 compounds in vitro at a laboratory in order to confirm if they are CB1 agonists. Thank you for listening.